Um, I just spent the last half hour kind of rushing through it. I got an email I missed a week or two ago about they're looking for volunteers for the dental board, integrated board construction. Never done that. That sounded like it'd be fun. Sitting there trying to get all this information in order to come here and lecture. I got it done just in the nick of time. And they'll let me know in October. I'll have forgotten I ever signed up for that. Okay, so today I'm supposed to talk to you twice this morning and then fortify yourself with lunch, thinking about digestion as you do so undoubtedly, and then this afternoon. Now neither of these lectures is real big, so we'll just see how it goes, and if it goes maybe a little faster than what I expect, then we might not have to do this afternoon. Another disclaimer here is this afternoon is GI motility, which by rights I would have put at the beginning of this course, not near the end of it. But for one reason or another, and when people were available to give lectures, it just ends up, this is the second year in a row, it ends up near much later in the course. So we'll be telling you stuff that probably should have told you quite a while ago. First part, what we're scheduled for this morning, is to finish up how do we digest carbohydrates and proteins. We already covered the lipids. The lipids is pretty involved in emulsifying, digesting, reconstructing, and distributing through the body. So that was a pretty long story compared to what we do with carbohydrates and proteins. And just by looking at how much time it takes to talk about them, you can also get kind of a feel for how quickly can we digest them. So it takes a lot longer to digest lipids and absorb them and distribute them than it does to digest carbohydrates and proteins. Now these two, we can really digest carbohydrates quickly compared to even proteins. So we'll start with carbohydrates, and when we eat or ingest our dietary carbohydrates, and if you look at what typically you might be getting in your diet would include starches, and here's part of this is kind of national board terminology that you might want to know. So starch, if you go back to FLS, we have two types of starches, two main types. Amylose and amylopectins. The difference between amylose and amylopectins are amylose are long straight chain polymers of sugar, and amylopectins are branched chain polymers of sugar. And when I say sugar, keep in mind, and we've got the numbers repeated here somewhere, but we've talked about it before. In our diet, about 80% of all the sugars that we ingest in one form or another is glucose. Okay, so we eat starches, we eat glycogen, which is a storage form of glucose in animals. Isomaltose, which I found out leading up to Christmas. Isomalt is isomaltose, and that can be used to, you put it in solution, you melt it down, and then you can use it to glue together gingerbread houses. So there's an important thing for you to know. It dries pretty clear. Maltose, lactose, sucrose, cellulose. Cellulose, so when you eat that, that's going to be in plants that we eat, vegetables, and cellulose is one of those carbohydrates that we can't digest well or can't digest much at all. Why is that? Why can't we digest cellulose? It becomes, that contributes into the fiber in our diet. Why don't we digest it?
It's linked by beta linkages?
Yeah, we've got beta linkages or beta bonds, beta glycosidic bonds. Okay. Oh, and it's right there. Cellulose is not digested. You get the beta 1,4s. And then another quick review there. In things like glycogen, highly branched, or also in the amylopectins, we have alpha 1,4 glycosidic bonds, which produce long straight chains, and alpha-1,6 glycosidic bonds, which produce branch points. Quickly, why is it significant or important that glycogen is highly branched? What's the significance of glycogen being highly branched? It's our primary carbohydrate storage.
There's like more points to break it down when you need it,
Okay, or
or store it.
build it up during the good times. So the more endings that we have, the more enzyme molecules can be working simultaneously on one molecule. So we can, during the good times, build glycogen fast, and in the bad times, we can break it down quickly. Okay, so whatever we happen to have been eating at the moment, the carbohydrate digestion begins in the mouth with alpha amylase, which is also called ptyalin. And I don't know if you got to do it when you were, we had the opportunity in grade school where we would put a saltine cracker in our mouth. Anybody ever do that experiment? Perhaps the first experiment I ever got to do. I failed at it. I just have to take it that this actually happens. If you put the saltine cracker in your mouth, it begins to taste sweet after a while. Is that true? It just, I mean, it's obvious looking at it. When you put food in my mouth, it does not sit there very long. So the alpha amylase had no time to work. Okay, so carbohydrate digestion, alpha amylase, or ptyalin in saliva. Alpha amylase is active at neutral pH, so, and saliva has bicarbonate to maintain that neutrality. What does it do? Alpha amylase takes long oligosaccharides and breaks them into shorter oligosaccharides and also disaccharides. So the term oligosaccharide is referring to a longer chain saccharide. Disaccharide, that you have two sugars, two molecules. Now once you swallow that saltine cracker, that digestion by the alpha amylase stops, and it's because in the stomach we have a lower pH and that inactivates the alpha amylase. When we inactivate enzymes, we in this case, if we're denaturing enzymes, what are we doing to the enzymes? Another board question. What do you do when you are inactivating those enzymes? What's the low pH do? Denaturing them, but what does denaturation do?
You're changing the bonded.
Okay, and what kind of bonds? Hydrogen bonds and electrostatic bonds. What kind of bonds are not changed? Significant. Covalent bonds aren't broken by pH. Okay? So, nothing much is happening in the stomach, but once the stomach empties into the duodenum, then the dietary carbohydrates are exposed to pancreatic amylase. I don't know, I don't usually put alpha amylase in there, but pancreatic amylase. So pancreatic amylase is the secretion of the pancreas produced by the acinar cells of the pancreas. We saw that with the secretions. It can be active, so it's not a zymogen, it's active as it's being secreted, and it's active in a neutral pH. The stimulus for pancreatic amylase secretion is cholecystokinin. And what assures us why are we neutralizing the chyme? That's under the control primarily of the chyme from the stomach is acidic. The pancreatic amylase needs a neutral environment. CCK was stimulated because the stomach was emptying. CCK is promoting the release of the pancreatic amylase, but we need to take care of the acidity. We need to neutralize it, and that's primarily going to be under the control of what?
Bile.
Not bile. Bicarbonate from the pancreas, which is stimulated by secretin. Okay. So once the pancreatic amylase gets working, it's taking those long-chain oligosaccharides and the shorter-chain oligosaccharides that were being produced by the alpha amylase, and now its job is to break things down primarily to disaccharides. So when you think of amylase in general, kind of its optimal product is, or ultimate product, are disaccharides, and those, the three main ones we get would be maltose, sucrose, and lactose. And of those three, the predominant one we get is what? Maltose, because 80% of our sugars are glucose. So maltose is glucose, sucrose is glucose, fructose, and lactose is glucose, galactose. So two stops here, and we've gotten down to disaccharides. The problem is digested disaccharides are still too big to be able to get into the cell. Okay, so that's not what's absorbed. So we have one final thing to do, and that's to break the disaccharides, the maltose, fructose, sucrose, and lactose into simple sugars. And we have three enzymes produced by small intestinal mucosal cells and parasites, which can break them apart. Now, glucoamylase is one of those enzymes, but most commonly, the name I would use is maltase. What maltase does is break maltose into two glucoses. Then lactase breaks lactose into glucose and galactose, and sucrase breaks sucrose to glucose and fructose. Of these three, the one that you hear most frequently about is lactase, because people are lactose intolerant, and it's because either they were born with low levels of lactase, or they, with age, start to produce less lactase. So the final product of chemical digestion in the lumen of the digestive tract, final products would be glucose, representing about 80% of all the sugar, fructose, which is about 10%, and galactose, 10%. And in FLS, we saw that briefly, that whether you're getting glucose, which we know we take through glycolysis, or galactose or fructose, those two also feed into glycolysis. And if we add up the cost in high energy phosphate bonds used in trapping and committing the carbons to glycolysis, and look at the net yield of ATPs, up to 38 ATPs per glucose, we find out that fructose and galactose also will have a net yield of up to 38 ATPs. So all three of them are equally valuable as energy donors. So we've got these floating around outside our body in the lumen of the digestive tract. Next thing is we got to get them in. And we want to do that efficiently. So as we're breaking things down chemically, we're starting to see glucose levels, for instance, going up and up and up. And we might be tempted to think about having just diffusion allow the glucose to enter our body. That's not going to work real well, because glucose is hydrophilic, and the lipid bilayer of the membrane, luminal membrane of the enterocytes, is going to resist its movement. That, and we want to make sure that we're getting this stuff into our body. So we're going to use active transport, and over and above just using facilitated diffusion. But active transport, we're going to use sodium absorption from the diet as the driving force, providing the energy for the uptake. So that's true for the glucose and also the galactose. Fructose, I'm not as sure about how it works. Most likely requires a transporter. You get some bulk flow absorption, but probably requires a transporter. So we're going to focus especially on glucose. We need a sodium-dependent transporter, active transporter. So sodium absorption releases energy, which allows us to pump the glucose into the enterocyte. Okay? Now these hexoses, 6 carbon sugars, need to once again be transported out of the enterocyte across the abluminal membranes to get it into the blood, into the interstitial fluid, and from there into the blood. So we also have transporters from in the abluminal membrane to transport the sugars into the interstitial fluid. From there it enters the blood, and this is the hepatic portal system. So absorption is taking place where the hepatic portal system exists, and that means all of this dietary sugar that we just absorbed, first thing that's going to happen is the next capillary bed it hits is in the liver. And that gives the liver a chance, first shot at the sugars that we've eaten. And that's it for carbohydrate digestion. Next one, protein digestion. In the U.S., I think this is from a while ago. It's probably an underestimate even back then. But in the U.S., estimate around 70 to 100 grams of protein gets ingested per day in the U.S. That's about, we're taking in about twice as much protein as we need to. Now, why do we need to eat protein? Because it tastes good. Special mammalian protein. You don't know why you eat protein? Or you all agree it's just because it tastes good? Everybody eats protein. No, I just had a flashback to my little grandson who was saying, everybody poops. They teach them GI physiology too. So why do we need to eat protein? Why do you need to eat fat? The answer is going to be the same. Huh? No? Carbohydrate can give us energy. And we could pretty much we could get by without eating carbohydrates.
Rebuild
Huh?
amino acids,
To,
essential amino acids.
yeah, we need the essential amino acids. What are they? Private Tim Hall. Yeah, that's all we need to remember for the board. Okay? Why do we need to eat fat? 2 reasons. Huh? Yeah, we could get by without the energy. Jack Sprat could eat no fat, but we need to eat fat. Huh? Yeah, 1, we need to get the fat-soluble vitamins. So if we cut fat out of our diet, we should start to exhibit deficiencies in what vitamins? A, D, E, and K. You're ready for that one. Okay? What's the second reason you need to eat fat? Tastes good. Bacon, just everything you need. What's the second reason you need to eat fat? No? I thought you'd get this one before you got the A, D, E, and K. Because of the essential fatty acids. What are the essential fatty acids?
Linoleic.
Huh? Linoleic acid, linoleic acid, and because they can be used to make arachidonic acid. Okay, now we got that. So we need protein to replace catabolized amino acids, specifically the ones that are the essential amino acids. Thought of another question here. Carbohydrates, so if 10 amino acids are essential amino acids, trying to think of how to ask the question. Okay, we'll start with this one. How many amino acids are there? No. Lots. I don't have a number for it. Lots. But why would you think 20? Because those are ones used for protein synthesis. Okay, for instance, proline is something used for protein synthesis. But we can also see an amino acid called hydroxyproline. Where do we see that? In collagen. Okay? But it's not used for the synthesis of collagen, it's the result of a post-translational modification. So, there's lots of amino acids. We kind of focus on these 20 that we need for protein synthesis, 10 of which we either can't make or can't make enough of, so we need them to get them in the diet. The other 10, we can make enough of them. We can get by. And what is the carbon source for those 10 non-essential amino acids? That we can make them. Now this is evolving. The questions are evolving in my mind. What's the carbon source for non-essential amino acids? So this is a release board question that I just typed up yesterday.
Alpha-ketoglutarate.
We can use alpha-ketoglutarate to make what? What amino acid? Oh, this is the easy part now. Huh? Glutamate. And if you can make glutamate, what other amino acid can you make from it? Glutamine. Glutarate, glutamate. All works out. Okay? But where are you going to get the alpha-ketoglutarate from? Huh?
Pyruvate?
Oh, you can get it from pyruvate. What else? If you took pyruvate and you add an amine group onto it, what do you have? Huh? Alanine. Another amino acid, a non-essential one. Where are you going to get your pyruvate? Huh? From glucose. Okay? So, what's the source of carbon for the synthesis of the non-essential amino acid? Yeah, carbohydrate. So another reason for eating your carbohydrates would be to make the non-essential amino acids obtained. What happens? What do you get if you take oxaloacetate? Where's oxaloacetate? Where do you find it? The citric acid cycle. What would happen if you took oxaloacetate and stuck an amine group onto it? What would you have? No. Huh? I thought you said Spartacus.
It's acidic, so
Yeah.
it's gonna be either too toxic.
Aspartate. Yeah, so down here, this is the TCA cycle, using my LA lesson. Down here about 5 o'clock is alpha-ketoglutarate, and we can make glutamate from it. Up here about 11 o'clock, half of the dial across the way, is oxaloacetate. Add an amine group to it, you get aspartate. The two acidic amino acids are 6 hours apart in the TCA diet. Okay, so we eat more amino acids. All of that triggered from we eat more amino acids than we need to, or proteins. So what do we do with the proteins? First thing we're going to do is we're going to chew them, and so we've got physical maceration of it. But most digestion of protein, chemical digestion, begins in the stomach with proteases, and then other proteases are added on by the pancreas and in the small intestine. That's going to finish the job. So So what's gonna happen? Chew our food, break it into smaller pieces, and then it goes down through the esophagus, into the stomach, hits the stomach, we got low pH in the stomach, and that's going to help to denature those proteins. Okay, let's make them non-functional because we don't know what proteins we may be taking in. So if you're eating a piece of red meat, what are the primary proteins you're taking in? You're taking in lots of different proteins, but what are the 3 most abundant proteins you're going to be ingesting when you eat your hamburger or turkey burger? This you'd be pulling for people who have had physiology before, for 2 of the 3. You should all know this from this biology. What are 2 abundant proteins that you find in skeletal muscle?
Aluminum.
Proteins. What does skeletal muscle do? Huh?
Actin and myosin?
Actin and myosin, yeah, for those contractile elements, right? Now, I said 3, what else would be pretty abundant muscle? And then got the actin and the myosin, now I gotta think outside the box or outside the cell. Huh? Troponin, yeah, we'll get some, they're little. Something we already talked about. Nah, we didn't talk about it today. Yeah, we talked about it actually within the last 5 minutes. Huh? Collagen. Collagen's helping to hold all these muscle fibers together. Okay, so that part doesn't matter much, but just kind of fun to think about. So low pH, the stomach, we denature the dietary proteins. Then, in the gastric secretions, digestion of dietary protein really begins with pepsinogen. Well, actually, it doesn't begin with pepsinogen because pepsinogen's a zymogen. It needs to be converted from its inactive to its active state. The active form of pepsinogen is pepsin, and pepsin is, if we want to go all fancy on it, an acid-stable endopeptidase. So it's been secreted, and it's a peptidase, and it's stable and functional in low pH. Now, what converts pepsinogen to pepsin? The very first conversion begins because of the low pH. So in acidic conditions, pepsinogen becomes somewhat unstable, and it fractures, it breaks. We break off a covalent bond, and one fragment becomes a pepsin. So we need, what's the activating compound? It's hydrogen ion. Once you have pepsin, it's a peptidase, and it can convert more pepsinogen to pepsin. So once you get a little pepsin, you get a lot more. Okay, but you need the low pH in order to start the process. What does pepsin do? It takes long polypeptides and breaks them into shorter chain polypeptides. And along the way, we may get a few amino acids. But basically, what pepsin is doing for proteins is what we saw alpha amylase was doing for carbohydrates. And then taking and making shorter chain links, but not accomplishing much beyond that. Now, after that's been rolling around in the stomach, the secretions have been coming out, the pH of the stomach has been dropping further and further, and we've had some time for the pepsin to start breaking apart these proteins into smaller pieces. Then the chyme gets released into the duodenum, and in the duodenum we see pancreatic bicarbonate neutralizes the chyme, which is going to tend to make the pepsin no longer functional. However, the pancreas is also secreting a number of other potential proteolytic enzymes. And these include, and they're zymogens, dangerous to make these things. Zymogens, it includes trypsinogen, chymotrypsinogen, procarboxypeptidase A and procarboxypeptidase B, and proelastase. Elastase is going to be breaking down, converting proelastase to lastase is going to help to break down the extracellular matrix. Okay, so just like we saw in the stomach, we have these inactive precursors, these zymogens, which need to be activated. And the activation process takes place in the lumen of the small intestine due to an enzyme called enterokinase. Entero for small intestine. Kinase, I don't know. Whenever I see kinase, I figure ATP must be involved. But this is extracellular, so I don't expect to see much ATP floating around. So enterokinase, another name you may see used for it is enteropeptidase. Now, this is the one that I learned. This name tells me more. Kinase gets me confused, but entero for small intestine and peptidase, that it's going to break peptides. So what happens is, this is on the luminal membrane of enterocytes, the outer aspect of the luminal membrane of enterocytes, and it is pretty specific for trypsinogen. It's a peptidase, so it's going to clip trypsinogen, breaking it into two fragments. One is trypsin, and the other is the ogen. So the trypsin is functional. So we've made an active proteolytic enzyme, trypsin. And just like we saw with pepsinogen in the stomach, once we have some trypsin, it can activate more trypsin. It's the one that activates the chymotrypsinogen to convert it to chymotrypsin. Trypsin is the one that converts procarboxypeptidases to the carboxypeptidases, and it's trypsin that converts proelastase to elastase. So once you've got some trypsin, you're going to get a lot of active trypsin and the other peptidases. And that's why enterokinase or enteropeptidase is located far from the pancreas. Because once you get a lot of peptidases, you want to have it in a protected environment where it's going to be working on the dietary protein, because we've got this sheet of mucus protecting, lining the wall of our digestive tract. Okay, so what are these different proteolytic enzymes going to be doing? Each one of them, it has the ability to break certain peptide bonds between this amino acid and that amino acid. It's been identified. Some of them can do several different types of peptide bonds. Others are pretty specific for one. You can look them up. You don't have to memorize them for me. Just know that they can act at different places. So they're going to go in, they're going to take these polypeptides, and one of them will start to act to divide it into oligopeptides, shorter length polypeptides, and then the others are going to keep acting on theirs. And so what we're gradually doing is kind of dicing this polypeptide chain up into shorter and shorter fragments. Doesn't matter which one hits first. It's just over time, it's like we each have a knife and we're hitting certain peptide bonds. So when they get done, and this is taking place in the lumen, in the small intestine, we're getting very short chains, so oligopeptides and even shorter chains. Now there's another enzyme that's being secreted by enterocytes, and these are called aminopeptidases. And what the aminopeptidases do are they're going to go in and they're going to, once we're getting down to shorter and shorter lengths, they're going to be clipping off single amino acids, and they act on the N-terminal, the nitrogen terminal, of these oligopeptides. And so the aminopeptidase is finishing off what we can do on the outside of our body, in the lumen of the digestive tract. So when that gets done, the aminopeptidase gets done, what we have is a bunch of amino acids. Okay, that's the final product of gesundheit, a fully digested protein. But in addition, we have dipeptides and tripeptides. So 2 amino acids linked together, 3 amino acids linked together. Anybody know what's a dipeptide that, especially like a year ago, was making the rounds on social media? A dipeptide suddenly was supposed to be bad for you. NutraSweet. What's NutraSweet? It's artificial sweetener, which consists of, it's aspartame is the chemical name. It's I forget what it is. Phenylalanine and aspartate, I think it is. People who have, they've known since the very beginning, people who have phenylketonuria can't digest that well and can cause them problems. But then all of a sudden on social media it's like, oh no, aspartame. It's dangerous, it's just 2 amino acids. Okay, so end product of digestion of peptides outside of our body is single amino acids, dipeptides, and tripeptides. Now, we need to get these things into our body, and then we need to get them into the blood. And the first part is going to be getting them into the body, and we have a whole family of sodium-dependent active transporters, okay? And those transporters, some are pretty specific for certain amino acids or similar structured amino acids. Other transporters can handle the dipeptides and the tripeptides, okay? But it's a family of these, it's not a single thing that's doing this. And this is where we're entering the body. Now once the dipeptides, the amino acids get into the enterocyte, nothing has to be done with them. They're going to be transported across the abluminal membrane to get into the blood in the hepatic portal system. But the dipeptides and the tripeptides, once they're in the enterocyte, they get broken into individual amino acids. And those resulting amino acids get transported across the abluminal membrane and into the blood. So I have to be pretty careful here in the way I think about things. In what form do dietary proteins enter my body? Partially correct. Yeah, amino acids, dipeptides, and tripeptides. If we shrunk ourselves down and we're sitting in the lumen of the small intestine, we're looking around, we'd see amino acids are being transported in, so are dipeptides and tripeptides. Now, that was my first question. In what form do dietary proteins enter the body? The correct answer is amino acids, dipeptides, and tripeptides. Now, here's another question. In what form do dietary proteins enter the blood? Amino acids. End of answer. Okay? So there's a final conversion of part of our dietary protein where we convert the dipeptides and tripeptides into amino acids. That's taking place inside our body, specifically inside the enterocyte. Okay? And then once they go in, first they enter the hepatic portal system and the liver gets first shot at them. And that's it for protein digestion.